Okay, welcome back, everybody. We're going to move on to our next talk. Um, Amy, are you ready?
Yes, I don't know why. I can't see myself, but can you see me?
I can't, but
Okay, wait.
as long as we can. Oh, there you go.
Oh, okay, here I am.
Perfect. Yeah, there it goes. Right. So, our next talk is from Amy Grogan, who is a PhD student in aquatic ecology at UNC Wilmington. Um, she's working in the aquatic ecology lab there, and her research focuses on water pollution, particularly harmful um, algal bloom ecology. The bulk of her dissertation research has focused on assessing harmful cyanobacteria blooms in the greater Wilmington area. She is also passionate about teaching and is currently searching for faculty positions. Um, Thank you so much for being here, Amy. We're very excited to hear what you have to say, especially someone from the coast, representing the coast well for our Western North Carolina group. And with that, I will hand it over to you.
Okay. Um, all right. Share screen. Let's just do.
Looks
Okay.
good.
Can everyone see the screen? The
Yep, looks
PowerPoint?
good.
Okay. All right, great. I'm Amy Grogan. I'm a PhD student at UNCW, and I'm going to talk to you about harmful algal blooms and particularly those that are happening in the coastal zone. So, one of the things that we're seeing a lot in the coastal zone, and I guess throughout North Carolina, is increased development. But I think it's probably the kind of the craziest here. And I'm going to talk about some problems associated with that with algal blooms, and then also about some potential solutions or mitigation strategies for those problems. So first thing, what is an algal bloom? We're probably familiar with that as water quality people, but I'm going to go over it a little bit. So massive growth of algal cells. Usually the dead giveaway is water discoloration. However, sometimes we don't see this very blatant water discoloration, and some of the other telltale signs are high saturation of dissolved oxygen and high pH values, usually above 8 in fresh waters. And then our state standard for what we consider an algal bloom is anytime that there's chlorophyll A content in the at or above 40 micrograms per liter. So, who are the bloomers? The big one that I'm going to talk about and that we see is cyanobacteria. So, that's very problematic. Some of the others are chlorophytes, and then kind of a mismatch group of others that are occasionally around euglena, chrysophytes, and cryptomonads. And then where are these happening? In primarily urban water bodies. So ponds, specifically stormwater ponds, or any area that receives runoff, so creeks, lakes, residential areas. And why are these blooms so problematic? So we know a lot of this stuff. As blooms decay, they increase biological oxygen demand, causing a crash of dissolved oxygen. Um, they impair the health of ecosystems and aquatic organisms. They are sometimes toxin producing, and the big toxin that is associated with cyanobacteria is microcystin, but there are a couple others as well, but this is kind of the big one. Sometimes they're associated with odor and they can cause the closure of recreational areas. And something I'm going to talk about in a bit of a case study about one of the sites here in Wilmington is that the occurrence of these blooms can add to internal nutrient cycling. So the thing that we're probably more familiar with is external nutrient availability or loading. And how is that happening? We have our gateway pollutant, as I like to call it, stormwater. So, what do algae want? Light and nutrients. Where are they getting it from? The light, not so much from stormwater, but they're getting these nutrients from stormwater runoff, specifically inorganic phosphorus, inorganic nitrogen. Fecal bacteria is also being loaded into the system from stormwater runoff, which as fecal bacteria break down, more nutrients are going to be released. And then TSS, which is mostly, particles of various things like dirt and sediment, which also can have nutrients associated with them. So where are they happening? Ponds, lakes, creeks, all areas that are potentially affected by stormwater runoff if they're a receiving body. And stormwater runoff becomes more problematic the more impervious surface coverage you have in your watershed. So areas like sidewalks, roads, any development, anywhere that's not a natural area where water can filter through dirt or the environment, you have an impervious surface, which means no filtration before this water enters its receiving bodies. So I'm going to kind of talk about two of the aspects of my dissertation work. And this one was kind of a big sampling of blooms across the Wilmington area. So here you can see all of my sites. And I sampled about 40 different sites. And those were in primarily isolated water bodies like ponds, the blue. But oftentimes, especially on the coast, these were near or adjacent or in the watershed of tidal creeks. So that means that these problematic blooms algal cells or toxins could potentially spread into the estuary or larger environments, larger water bodies. In the case of Greenfield Lake, which I'm going to talk more about later on in the presentation, the outfall of this lake where lots of blooms happen is the Cape Fear River, a very important water body, and also the public drinking water source for the city of Wilmington. So when you're starting to do your sampling, and when I was doing my sampling, something that's really important is to identify your sampling locations and your hotspots, right? So places where there's high impervious surface coverage in the watershed, areas that are warm, stagnant, and shallow. The water, again, a lot of these ponds, so you can identify where is the problem and how bad is it. And over the course of this sampling, over the course of 3-4 years, I sampled about 90 algae blooms, but you can see that there were only 40 sites. And that's because there were a lot of repeat offenders. So usually what happens is once you've identified a place where one bloom is happening, you're going to get consecutive blooms. And that's kind of that internal nutrient cycling that I'm going to talk about. So sampling methods. Sampling blooms as they're observed is very important, but that is not always as feasible for consistent monitoring programs like many of the municipalities have. So if you can have ongoing sampling, in Wilmington, we have ongoing Wilmington watersheds research that we do through my lab. So we know areas where there are pollutants in the water or that have high eutrophication that might or likely will end up having algal blooms. So both of those things are important. Then identifying your taxa that are present in your algae blooms. So here there's actually a flagellated chlorophyte here, and then cyanobacteria. Some of these cyanobacteria can be kind of tough to tease out, and this has been a real learning curve for me because my background is in more marine stuff prior to getting to the dissertation, but I've gotten real good at teasing out these cyanobacteria. So identifying your taxa, the more cyanobacteria that you have, the worse the situation is, because again, they can produce these toxins. So when you do have cyanobacteria, an important aspect of sampling is to test for the presence of toxins. And the big toxin that these guys produce is microcystin. So the way that I tested for this, the very simple way to test for this is that there are commercially available test strips. And actually, with the unfortunate advent of COVID, these test strips work very similarly to the COVID tests, which is kind of interesting. But they're very simple to use, great instructions, easy to do, and easily accessible. A little expensive, but easily accessible to, lots of people. So that will give you an idea of how much toxin is in your water, and you can go from there to try to assess which toxins are present, but that requires some more detailed chemistry and instrumentation. And then the other thing that's important to do is to determine what the concentration of your bloom is, or what the intensity with your chlorophyll A concentration, and determine what kind of nutrient dynamics or nutrient concentrations are in the water, because that's going to help with identifying what the predictive factors are, and then putting mitigation strategies into place based on what these factors promoting blooms happen to be. So in my sampling, I delineated blooms into type, location, and taxa. And I had 4 different types of blooms that I broke it down into. So water column distributed, which here you can see that nice green color, but sometimes these can be a little more difficult to spot, which is, again, the importance of taking those other measurements to try to identify blooms, because the cells are water column distributed and we don't have this very clear scum at the top. So surface scums, we definitely know there's a bloom going on there, right? No question. And this is actually in a city park, so that's not good. And then surface mats. So these are primarily of filamentous algae. And then finally, benthic mats. Sometimes of filamentous chlorophytes. So always, yes, filamentous algae in the mats, but mostly the surface mats chlorophytes. And then the benthic mats were usually a mix of chlorophytes or some of the benthic filamentous cyanobacteria, which can produce toxins. So water column was the most common. And then locations, ponds, lakes, and creeks. And then the taxa. So cyanobacteria, chlorophytes, and euglenoids were the most pervasive. And then there were some others in the mix. So a lot of others, actually. Dinoflagellates, even in freshwater. Diatoms, chrysophytes, and cryptomonads. And when you do identify your taxa, it's really important to successfully identify and do further analysis when you have your problem children present, those that can produce toxins. And one of the big ones with toxin production is microcystis. So this whole top row here are actually different types of microcystis. So see what I mean about trying to tease out the taxa. These are all the same genera, but all different species. But when you really look at them in the microscope, they all look the same and the descriptions all sound the same. But anyway, this is all microcystis, and that is what the toxin microcystin is named for. So the taxa along the bottom here are also capable of producing microcystin, but in my experience, microcystin was identified when microcystis was present. So when you're seeing these microcystis species in your water, that's definitely a red flag. Others down here, Dolichospermum, Aphanizomenon, and this one is kind of a newer kid on the block, if you will. This is Cuspidothrix, which was not thought to be in North Carolina or I think the United States until recently. And this algae actually we found in blooms that were producing microcystin and saxitoxin. So that's kind of another big thing coming up in the kind of, I guess, toxin, algal toxin sampling. Microcystin is kind of the one that's the go-to for cyanobacteria, but we're finding a lot of them can actually produce saxitoxin too, which is the paralytic shellfish poisoning toxin that's usually associated with marine genera. Okay, so what did I find in this broad sweep of Wilmington? Most of these blooms were water column distributed, cyanobacteria, and they were occurring in ponds, these stormwater ponds primarily. And what were the drivers of these blooms? So water temperature was a big one. Again, in these stormwater ponds, they're shallow, they're usually in full sun, and they're getting very warm. So our blooms were occurring more at the higher temperatures above 25, and that was all types of blooms. But if we break it down from 25, below 25 to above 25, what we're seeing is in the lower temperatures, this is where we have more chlorophytes and our other taxa. So all blooms are not ideal, but the cyanos are really more problematic. So lower temperatures, not as much of these cyanobacteria, but when we're getting into the higher temperatures above 25, this is where the distribution of cyanobacteria is the largest, and that's where more of the cyanobacteria blooms are happening, and the other types are kind of coming down and being less frequent. So with climate change, warmer temperatures earlier in the season, longer, we're seeing more of these cyanobacteria probably happening. And warm temperatures are also associated with some nutrient cycling. So something that was also a big driver was dissolved inorganic phosphorus. All blooms, all types of blooms were more intense or had greater chlorophyll A concentration when there was higher phosphorus in the water. And in this upper corner here, where the phosphorus was the highest, we see that we have the most abundance of cyanobacteria. So we have the little green triangles are cyanobacteria. So most of that, the cyanobacteria was happening in the highest phosphorus concentrations, and the most intense cyanobacteria blooms were happening there. And the increase of phosphorus in the water was also associated with the presence of microcystis, the actual algae, and then microcystin, the toxin. So how are we getting from these earlier blooms and greater dissolved inorganic phosphorus to these harmful cyanobacteria blooms? So we know that nutrients are coming in from stormwater, and those can support algal blooms of any type. And the more of that you have, the higher intensity your bloom is going to be. But how do we get to here, this really high phosphorus situation and this dominance of cyanobacteria that are potentially producing toxins? So the sediment acts as a really large reservoir of nutrients and phosphorus in this case. So I'm going to talk more about some other nutrient cycling in the sediment and how it acts as a reservoir. But when we have our stormwater runoff coming in that's rich with dissolved organic phosphorus and nitrogen, if there's not algae or other bacteria in the water to process and uptake that, then it's going to end up in the sediment. And that happens more often in our cooler months when we have less light. But stormwater is happening all the time. Eutrophic stormwater always getting added to these receiving systems. So if something's not taking it up, it's ending in the sediment and just sitting in a pot, right, waiting. So we get these algal blooms occurring. When algal blooms happen, we have decay following the bloom. And then we also have, in the summer months, increased temperature, which is also, and light, which is also going to promote more blooms happening. And both the decay of cells and the increase of temperature is going to cause our dissolved oxygen to lower and potentially crash, getting into a hypoxic situation. And once we have hypoxia, no oxygen situation, the phosphorus that was happily asleep bound to the sediment gets released from the sediment because of the change in oxygen state. So when it's oxygenated, it stays bound to sediment. even in the warmer weather or what have you, when algae is present. But once we get to a low or no oxygen state, that phosphorus is released. So now we have an external source and an internal source. And now we're getting more cyanobacteria, more intense blooms, and more toxin production. So bad news. And in this study, I found a diverse number of different cyanobacteria. So in this wheel of fortune here, all of the warm colors are different types of cyanobacteria. And the way that this wheel works, it's not, the 3% is not, oh, there was 3% of this one, 3% of this one. Essentially, the way it calculates these percentages is based on, okay, there was, this is the easy one to do. Chlorophytes made up 11% of all of the total genera identified. So each one is 1%. So this isn't the distribution of how often the individual taxa were there, but the distribution of, you know, higher classes to lower. Hopefully that makes sense. Anyway, high diversity of cyanobacteria. So potential for toxin production. All of the ones that are highlighted in yellow are potential toxin producers. So 17 different genera of toxin producers were found in that short time. And this was just me doing this sampling. So if there was a team of people, it probably would be even more. And primarily, these were in the Nosticales class, which are our nitrogen fixing cyanobacteria. So again, it's important to test for toxins. Uh-oh, it says my internet is unstable.
Yeah, Amy, you just cut out for like around 10 seconds, I'd say.
Okay.
Mind going back
Am
a little
I
bit?
back in?
Yeah, you're good now. You're good now.
Okay, that was strange. I'm at home, so um okay. Well, I was talking about, yep, the different algae, different cyanobacteria, lots of different toxic cyanobacteria. The importance of when you find toxic cyanobacteria, testing for microcystin or other toxins. But the thing here in my study, my broad sweep of the Wilmington area, I did not find a lot of incidences where toxin was actually being produced. However, I was only testing for one toxin type. And if you look at this table here, these different cyanobacteria genera, all found in the study, produce a number of different toxins that are not microcystis. So, or excuse me, microcystin. So microcystin is the MC, and then everything else that's on here is another toxin that the genera is capable of producing. So one of the big take homes here is that it's more than microcystin. And if possible, it's important to test for as many of these toxins as possible. And if not, to consider any genera that's capable of producing toxins, not necessarily toxic, but treat it as though it might be, because unless you tested for it, you don't know it could be there. All right, so where are the hot spots? Stormwater runoff areas that are receiving eutrophic stormwater runoff, where you have external nutrient loading, stormwater ponds, and other stormwater receiving water bodies are going to be hotspots for these blooms and good spots to try to start sampling. And then areas where you have stagnant, low to no flushing, shallow, and hot or warm waters. So again, these stormwater ponds, really big spot for these. So that was kind of a broad assessment of blooms across the Wilmington area. And I'm going to talk specifically about Greenfield Lake, which was an area where there were repeat offenses of harmful algae blooms. And it's a really good kind of model system for a worst case scenario, honestly. So Greenfield Lake is an urban lake and a city park in Wilmington. The outside of it is actually very lovely. There's lots of cypress trees and other beautiful native vegetation. But the water quality is very poor, unfortunately. And that's driven by runoff. So Greenfield Lake, although it is a lake, is almost like a giant stormwater pond because there is stormwater runoff from a huge watershed that's highly impervious or has highly impervious surface coverage coming into the lake. So harmful cyanobacteria blooms occur annually in the lake. And in 2022, for like 2 months straight, the whole lake was in bloom, and that's not uncommon. So this next kind of information is an assessment of bloom samples and then also ambient water samples. So Greenfield Lake is a place that we know is problematic, and there are actually some mitigation strategies in place that I'll talk about a little later that we do regular sampling at. So this was part of the Wilmington Watersheds program, and doing sampling regularly here is beneficial because again we can make appropriate mitigation strategies to try to prevent these things from happening. So this is also going to highlight the importance of both the internal nutrient loading and the external nutrient loading. All right, so here these are the results of a prediction tree analysis from the samples from Greenfield Lake. It looks kind of complicated and crazy, but essentially what it's saying is that the numbers, the lower the number is, the higher importance the factor is in predicting the presence or absence of blooms. So in this case, temperature was the biggest predictive factor. So temperatures greater than 24.5 were where most of our blooms were occurring. And then at lower temperatures, nitrogen, specifically ammonium and dissolved inorganic nitrogen, became important predictive factors. But in the bulk of our blooms that were at high temperatures, we actually had very low dissolved oxygen content. So high temperatures and lower, not dissolved oxygen, dissolved inorganic nitrogen. So lower nitrogen meant more blooms. So what's going on here? Why was the nitrogen so low? We know that these blooms need phosphorus, but they need nitrogen too. So we look back again at this wheel of fortune. We have these Nosticales as the most abundant cyanobacteria. And the Nosticales are very hardy and have great growth abilities because they can fix nitrogen from the atmosphere. So nitrogen is not as important to them, and therefore they're very pervasive, and they can produce toxins. So a couple of the things that they have that help them grow, but also be resilient and essentially create a seed population once they're present in a water body, are aconites. So that is a resting cell. That's the cell right here. And actually, just real quick, the darkest cells here are all vegetative cells. This is the heterocyst that I'm going to talk about, and this is the aconite. The heterocysts and aconites are not always present. Heterocysts usually are, but the aconites not as often. And that's because aconites are formed when water conditions are not as favorable, because they're a cell that forms cysts. So once the water conditions are not favorable and the algal cells die, the aconite can enter the sediment and hang out there until the conditions are favorable, and then it can germinate. And now you have algal, you have cyanobacteria that was not present over the winter that kind of makes its way up into the water column once the summer months come and it warms up. These Nosticales also have aerotopes, which are these small gas vesicles inside the cell. They're a little challenging to see, but these little dots essentially are the aerotopes, and that helps with buoyancy so they can be close to the light source. Very important in Greenfield Lake too, because it's actually a black water system. They have heterocysts, which are how they fix nitrogen. And what nitrogen fixation means is that typical labile nitrogen sources like ammonium, NH4, and nitrate, which is NO3, that other phytoplankton need in order to have a nitrogen source for photosynthesis is not something that they necessarily need, because they can take dinitrogen, which is in the water, but also atmospheric nitrogen, which we know that there is ample supply of, and convert it into the bioavailable form ammonia that they can then use for photosynthesis. So if you're having no nitrogen in your water when you're sampling your blooms, it could be that nitrogen was taken up by the algae at that time, but it also could be because it's not as important for it to be there because you have these nitrogen fixers present. However, another driver is nutrient cycling. And what we can, the nutrient cycling is kind of driven by that internal nutrient load and succession of blooms. So this kind of succession pattern in blooms throughout the warmer months in eutrophic systems like Greenfield Lake is pretty common. What we start with are usually filamentous chlorophytes, so the mat forming chlorophytes that look like this. And then we get into nitrogen fixing cyanobacteria, and then finally cyanobacteria that are not nitrogen fixing, that are the most associated with toxin production. So you can see that here, the light green are filamentous chlorophytes, the blue are the cyanobacteria that are non-nitrogen fixing, and then the teal is the nitrogen fixing ones. And that's not exactly the pattern in each of these, but this is by different sites across the lake. So this succession from one algae bloom type to the next is driven by the internal nutrient cycling. So nitrogen cycling within the sediment, and then sediment water interface nutrient dynamics. And what essentially happens is a feedback loop. So like I said, finding those repeat offenders, because once you've had one bloom, you're probably going to have another bloom. And it might start as non-harmful genera, but then move into harmful genera. So here's a kind of busy schematic, but this is kind of showing or it is showing the nitrogen cycling that's happening. So we have an algal bloom, and as it starts to die or senesce, we get the release of nitrogen. So in this case, we have ammonium. And when there's lots of that in the system, as it gets near the sediment, and we have this sediment water interface, similar to the air water interface, gases, and in this case, nutrients, are going to, go back and forth to be in equilibrium. So when we have all these cells dying and all this nutrient released, it's going to influx into the sediment. Once that's the case, we can have several nitrogen cycling processes happening in the sediment. Nitrification, which is going to take this and turn it into nitrate, a bioavailable form. Denitrification, which is going to take this ammonium and turn it into dinitrogen, but then also nitrogen fixation, that's going to take dinitrogen and turn it into ammonium. And eventually what happens is, as you have an algal bloom, it dies, then you have no bloom action going on. You have very limited nitrogen available in the water column. The nitrogen pools into the sediments. It starts cycling, it's creating more nitrogen, and the nitrogen that's no longer available in the water is outfluxing out of the sediment and spurring the growth of a new bloom. And when we have these nitrogen fixers, they have this nutrient source from the sediment, but also this nitrogen source from atmospheric nitrogen. The other thing that's going on here, we can see low oxygen. And if you recall from the other schematic, when our oxygen levels go down, the phosphorus in this system, in this reservoir here in the sediment, also gets released. So you get kind of a boom and bust going on throughout the summer months. So that importance of, even though we have external sources coming in, which are bad, obviously, once the more of that runoff that comes into the system, the more eutrophic runoff, the more the system itself holds on to that. And we have a problem with both external loading and internal loading. So some ways that we can try to mitigate this. Sampling, of course, is very important because you don't know if you have a problem or how pervasive or large it is until you start sampling. City property is a great place to start, and trying to identify areas that receive runoff. And then recreation areas, very important spot to sample because, you know, people might be interacting with the waters there, and pets. Stormwater sampling, also important because, again, runoff is the original, the OG pollutant, the gateway pollutant, and it's going to be driving the initiation, at least, of these blooms, or the initiation of this storage of nutrients that then drives the blooms. Another important factor is education. So signage is very important because many people are not aware that the water can be dangerous. So in Wilmington, I believe it was in 2019, and probably people are familiar with it because it made national news. There was a death of 3 dogs only hours after playing in water in a stormwater pond in someone's neighborhood. And it was probably from anatoxin, which is a very potent toxin, and there wasn't an actual surface bloom present. So this person had no idea, and that could have happened to a person or a child. So having these signs around to try to educate people and warn them is very important. Also, educating about the issues at large. So in Wilmington, we have this great nonprofit group called the Cape Fear River Watch. They do a lot of education in the area with citizens and kids, and doing work with schools is great. You know, the more people know, the more they'll care. And healthy wetlands, beautiful wildlife, happy people. Getting citizens involved in monitoring is also great, or in cleanup actions. There's a lot of that goes on in the coast with like living shoreline stuff, if anyone's familiar with that. So that's the planting, or, you know, moving oyster bags, or planting spartina grass to try to help the coast, and people, they get a lot of people involved in that. So you can do similar things in these freshwater systems, in the wetlands. Also, better planning. Proactive is better than reactive. And one of the biggest things, especially with the massive and very rapid development that's going on in Wilmington and coastal areas, and perhaps is the case in the Piedmont area as well. I'm not really familiar with it, but stricter development codes and enforcement. Ponds are problematic. We know that this is not a stormwater system that's working, because this is where all of the blooms are happening. All of these toxins are, like PFAS and other heavy metals, are also accumulating. So stormwater ponds are not a good solution for stormwater runoff and stormwater retention. Wetlands are a much better place. Rain gardens or artificial wetlands. The difference between a pond and an artificial wetland is just the placement of some native plants, and the, you know, kind of, instead of just a ditch, some structure. It's not that hard. They can definitely, developers can definitely do that. And then more pervious surfaces rather than impervious surfaces. This is a really nice picture. This is at a local bank. They have pavers here in their parking spots instead of all pavement. And then as much protection beyond the enforcement that you can muster in areas. Again, education, the more you can get people to care, the more you can bring up the issues, the more likely you are to be able to protect places and put policies in place that will protect places in the future. And I think that is it that I have for you. So some conclusions. Sampling starts the solution. Finding hotspots and can aid with hotspots for blooms is going to help you with ecosystem repair and figure out where the ecosystem impairment is. Identifying taxa and potential toxins, potentially identifying succession trends, doing some more involved sampling with nutrients and in both the water and sediment. So you can identify predictive factors. The more data, the more mitigation strategy or the better informed mitigation strategies you can put into place. Stormwater nutrient control. Again, the external loading is what then drives the internal loading, and focusing on both nitrogen and phosphorus, because a lot of the focus has been on phosphorus. And then better planning and regulations and informed community members is a great way to start your mitigation strategies for making sure your water bodies are clean and healthy. And that is really all I have for you. So I'm happy to take any questions, and I'm very happy to be able to have given this presentation to you.
Thank you so much, Amy. Now it's time for questions. Please post your questions in the chat if you have any. In the meantime, I came up with some. First of all, your graphs and diagrams, I really like those. Like
Thank
that
you.
wheel of all the different cyanobacteria, that was cool. But yeah, wow, Greenfield Lake looks just really bad.
Yeah, it's Greenfield Lake is a tragedy for sure.
Yeah. But yeah, good to know that we have people like you figuring out how to fix it.
Trying.
Yeah. Shannon Simpson says, fantastic presentation. This was incredibly informative and interesting and useful.
Thank you.
I would agree. I also want to say, I really liked what you said about signage too. I didn't realize people would let their dogs swim in a stormwater pond. But then I thought to myself, well, I didn't know what a stormwater pond was different than a regular pond before starting this position. So it is really
Right.
important to put up those signs. I agree.
And I mean, especially when there's not a clear like scum present in the water. People don't they see these ponds, they don't think, that that is potentially dangerous. A lot of times there's turtles and ducks in the pond. People go and feed them, kids go and play near them. So the importance of signage and informative signage, because there's actually a place where I sample where it doesn't have a sign like the one I showed. It just has a sign that says like no fishing and beware of alligators, but it's also an area where there's been several blooms and toxic blooms. I'm actually kind of doing a study on that right now, even though I'm trying to graduate. So I don't know why I'm doing that, what I'm doing to myself. But either way, and I see people fishing there all the time, even though there's no fishing signs. But I think they probably think that it's just one of those like, legal things. Oh, these people have to put these up because if they don't and somebody gets hurt or gets bit by an alligator or whatever, then they're liable. And I mean, I know lots of people think that, just like, signs that you see at the beach that are like, oh, you can't be over here, whatever. It's like, yeah, somebody just put that up because they don't want to get in trouble if something happens. So people don't realize that it's truly for their safety instead of like overreaching government, if you will.
Yeah, yeah, that's good to remember because I definitely don't want my dog swimming in a pond that has ammonia in it.
Yeah.
So yeah, that's really good to know. Especially part of my job is outreach based, and now I have another idea because sometimes, well, I haven't done it yet, but spray painting over storm drains, like no dumping.
Mm-hmm.
But yeah, putting up those signs too sound really important because yeah, we need people to stay safe. Um, okay, so you just said that there are sometimes turtles and other animals that live within these ponds that are filled with algae.
Yeah.
How is
Let's
how are they that resilient?
so if there's not toxins, I'm trying to here we go. I'll go back. Um some of them can leave. So here's a where's my birds? I see waterfowl all of the time in these areas. And actually, that's problematic. So there's some Canada geese there. So anytime you have a water body, waterfowl are going to flock to it. And that becomes a problem too, because the Canada geese poop a lot, and that also drives the nutrient cycling. And then the other thing is, when you first build a pond, where are these cyanobacteria coming from? One of the big thoughts is that the birds are moving them on their feathers and their feet. Again, talked about that really resilient resting cell, the aconite, or if they're in this water and then they fly over to a pond that's not far away, now they've taken this algae and moved it somewhere else. So usually, have not seen any dead birds. I have seen dead fish and I have not seen dead turtles. But one of the things with these toxins, a lot of times with microcystin, if it's not exceptionally high, it has more of a problem potentially with chronic exposure rather than acute exposure. So if these ducks and other things that don't have to necessarily breathe the water, they might be okay. But it gets to a situation potentially where, like with the dogs, what happened in the Wilmington situation was, and I'm gonna scroll over here to my toxin page. Where's that one? Microcystin is like the really common toxin we test for, but this one, they call it instant death anatoxin. So microcystin is kind of big, like similar to this one. And because it's large, it doesn't, it stays in the, or it stays in the water and doesn't break down, or I'm saying it backwards.
Like a longer half-life or something?
Yeah, it doesn't when it's released from cells, it can like hang around in the water. But this anatoxin here, when it gets released, it basically immediately gets broken down in the water. It kind of broken down. But it's normally in benthic things. So in the case of Wilmington, what kind of what they've decided is that these dogs were playing in the water. They either kind of ate or stirred up some benthic cyanobacteria. This anatoxin was released, and that's what they came into contact with. So some of these other toxins are more potent than microcystin, but microcystin is the one that is the most frequently tested for, and it's going to be the one that has the largest duration in the water. But cylindrospermopsin and saxitoxin are also larger molecules, so they can hang around too. I don't know if that answered the question.
No, that definitely did. So like kind of when you like dredge up the sediment at the bottom, it can release that anatoxin. That's what you're saying?
So the anatoxin is it's gonna have to like actively be in a cell. So yes, if you dredge up the bottom and there's live benthic algae cyanos growing on top of it, yes. But microcystin does have a long half-life, and it can actually stay the toxin itself in the sediment.
Gotcha.
Anatoxin, once it's in the water, it's basically gone because it's so small.
Right.
But it is the most deadly of them.
Wow. Wow. Well, that's
Yeah, bad news.
yeah. But yeah, we're at time. So thank you so much, Amy. It's been a pleasure having you speak at this conference. Like Shannon Simpson said, fantastic presentation. And right now we're going to take a 11-minute break until our final presentation of the day. Thank you again, Amy.
Thank you.
Thanks, Amy. That was great.